Okay, I wanted to touch on chapter 4, energy and metabolism. And these are the goals for this chapter. One of the things that we have to appreciate about energy and metabolism is that in order to live, cells need to ingest building blocks and an energy source. And that's what's contained in the diet, okay? Whether you're an animal and rely on energy from another source, or whether you're a photosynthetic organism and can create your own fuel and then utilize that as energy. Ultimately, you have to generate some form of immediate power in order for you to move and grow and reproduce and perform maintenance. And you have to utilize that in order to assemble the molecules that make up the cells and to promote any other type of work, right? This is a little sidebar here. Binge drinking is a serious problem on college campuses. I'm sure you're aware of this. ADH is an enzyme that helps break down ethanol and other toxins. Its breakdown products are very similar to the compound that preserves tissue, and what's called formaldehyde. Okay, so let me draw that real quick here. Okay, so it's a breakdown product. This is formaldehyde, right? Okay. Those are two H's, and that's a double bond O. That's formaldehyde. Okay. And what's produced when we break down alcohol is something called acetaldehyde, which looks like this. They have very similar properties. They preserve living tissue. Okay. But by doing that, they kill it. Okay. And the result is damage to the liver, such as cirrhosis, right? And then eventually what happens is you can lose liver function. And that will result in death, okay? So the breakdown products produce energy as well. Okay. But the problem is that the other byproducts are damaging. This is one of the reasons you don't want to engage in too much recreational drug and alcohol consumption. Okay, because if you lose your liver, you can't continue to function at all, right? So first topic here is just energy, which is the ability to do work. Okay, remember that when the universe was created, the three things that they were made of are matter, energy, and empty space. Okay, matter is anything that occupies space and has mass and comes in three forms: solid, liquid, and gas. Energy is the ability to do work, and empty space is empty space. Okay, work happens because we move energy from one form to another. Okay, we talked a little bit about this in class. Energy transformations happen all the time. If let's say this is a table, right? And let's say this is a ball on the table, okay? That ball, because of its height, has potential energy. Okay, and if I push that ball off the table and it falls, that flight to the floor, that is kinetic energy. Okay, And then some of that energy leaves as heat. Okay. So those transfers happen all the time. Okay. And then as heat, right? That's how the energy gets lost. And what does that do? That causes more entropy, which means the universe becomes more random. Okay. We talked about the first and second laws of thermodynamics earlier, right? The first law, energy can't be created or destroyed, but can be interconverted, right? There was the equation. Okay. So there's no magic, right? There's only so much stuff in the universe, and that's all you get. The second law of thermodynamics is systems seek the low energy state. Okay. And we talked about the fact that a system can be anything. It can be the dust on the table, the air in a room, um, um, putting sugar in a glass of water. Okay. Um, Eventually, it's going to find the low energy state any way it can, right? And those two laws went into effect when the universe was created. Living things maintain organization by getting energy from other places. Energy flows through the biosphere and then out into ecosystems. And energy in chemical bonds is a type of potential energy, okay? When those bonds are broken, they release energy that can be captured. Okay, we'll talk about exactly how. Okay, and of course, some of that is lost as heat. Okay, you can see an example here, right? We've got potential energy. This guy's dangling, right? Energy of position. Okay, and if he were to fall, that would become kinetic energy, energy of movement. Okay, right. So you can see here examples of how energy flows through ecosystems. We talk about the fact that sunlight leads to a process called photosynthesis, in which we take CO2 and water and we make sugars. This is glucose, and of course, there's going to be some heat given off. Okay, but this is now an energy source. Why does it have energy in it? Because UV rays, the sun. Well, let's be more specific. Red and blue wavelengths of electromagnetic energy from the sun drove this process, because these guys are low energy. And up here, this guy's high energy. And remember, I told you how you can tell if a molecule has a lot of energy, a lot of calories in it, it's by counting the number of carbon hydrogen bonds. There aren't any over here, and there's quite a few over here. Okay. The consumers eat what the producers make, and in a process called respiration, generate CO2 and water, okay, and heat and ATP from ADP and phosphates. Okay. And how much? About 38. Okay. So the consumers eat what the producers make. The producers also eat what the producers make, because the producers do both photosynthesis and respiration. But the animals, the bacteria, and the archaea just do respiration, unless you're talking about a photosynthetic archaea or bacteria. And where does all that heat go? Out into the universe. Makes everything more random. Okay. Cells store and retrieve energy in metabolic reactions. Some reactions require energy input and others release energy. A reaction is a chemical change, right? Chemical reactions involve the making and breaking of chemical bonds. Okay. A reactant is a molecule that enters a reaction, and the product is what comes out. So example, right? A plus B can combine to make AB, right? Or a reverse reaction, right? AB could come apart to make A plus B. Okay. Well, sometimes we can have AB plus CD producing AD and BC. Just different ways things can go. We can hook things together, we can bust things apart, or we can change dance partners. Okay. So this is an overview of how this works, right? Now, which way the reaction goes depends on the energy, right? We always go from high to low energy, right? Now, some reactions already have that built in, right? We go from high to low energy. Basically, we take something that's got a lot of energy in it, we tear it up, okay? And that's called decomposition. Okay. Sometimes we change dance partners, and those are called exchange reactions. Okay. And then sometimes we build things, and it's called synthesis. Okay. So over here, the energy is high, and over here, the energy is low. And where did the energy go? Other work and heat. Okay. Over here, the energy could be about the same. Okay. Equal energy. Okay. Approximately. Okay. Of course, heat is given off as well in either direction. Right? Then here, right, the energy here is low and the energy here is high, right? So how is this going to happen? Well, we have to steal energy from another source, okay? And of course, heat's also given off here, okay? So this reaction here is what a chemist would call not spontaneous. Doesn't happen on its own. And y'all know that just in your daily life, right? Things don't magically assemble themselves. This isn't the world you live in, is it? Right? But stuff falls apart all the time, and stuff can switch dance partners all the time. Okay? That happens out there in the world. Now you can see some examples here of energy considerations, right? Here we have photosynthesis. The energy in here is coming from the sun. Okay. So low energy, right? Down here, high energy up here, right? And then the reverse, right? You got high energy here, you tear this sucker up, you make these breakdown products, and the energy comes out, okay? And that energy can be used to do work, okay? And some of it becomes heat, okay? And again, we should also add that there's heat involved in this too, okay? There's heat given off in any chemical reaction, okay? So these guys down here are low energy, these guys here are high energy. Molecules of life release energy when they're combined with oxygen, but they don't do it spontaneously. The energy is required to start even an energy releasing reaction, which is called activation energy, right? And that's the energy needed to produce a collision between reactants. Okay. Now, that activation energy can be very high. Okay. High activation energy makes it difficult to start a reaction. Okay. Right. And a low activation energy makes it easy to start a reaction, right? Now here, right, we're seeing reactants and products, right? And this is going to be a spontaneous reaction. Once it gets going, it'll go on its own, okay? And it'll kick energy out, right? Here, from products to reactants, okay? The reactants are low energy, the products are high energy, meaning energy from another source. Okay. In order for this to go, okay? So we can draw an analogy here, right? If we have, let's say you got your car here. Here's my very bad drawing of a car. Okay, there you are in the car, right? And you got a hill, okay? And the object is you want to go up the hill. Well, if you sit there and hope that car rolls up the hill, it ain't going to happen. You all know that from experience, right? So how are we going to get that car to roll up the hill, right? We're going to have to pull energy from another source. And where does the energy come from? It comes from the gas in the tank, right? Gas tank, gas, okay? The gas in the tank gets sprayed into the carburetor, which mixes it with the air, which has oxygen in it. Then inside the engine block, that mixture of gasoline and air is ignited by a spark plug. That causes the pistons to move in the engine block, and that turns the camshaft, and that turns the crankshaft, and eventually that turns the wheels of your car, and then up the hill you go, right? So you took the energy of something that was a favorable reaction, right? Exploding gasoline, and you drove an unfavorable reaction, which is going uphill. Okay, so you stole the energy from something else. Okay, and the same thing is true down here in chemical reactions that drive life. Okay, so energy in, right, to assemble things, right? These are reactions that are called anabolic reactions. Means to build up. Okay, and then these reactions take a big molecule and tear it up. Okay, those are catabolic reactions. Means to tear up. Okay. So the anabolic reactions rely on the catabolic reactions to happen, okay? Catabolic feeds anabolic. Okay? This is why you have to eat to live, right? Now, we talked about the fact that for a chemical reaction to happen, reactants have to bump into things and make products, right? So there's several ways to do that. You can heat the reaction up, right? You could add more reactants. Okay. But both of these are toxic to the cell. Okay. Heating it up could cook the cell. Adding more reactants could cause water to come into the cell and blow it up through osmosis. Okay. So the way that we get around this in living systems is to use something called a catalyst, which speeds up a chemical reaction without needing heat or changes in concentration. That means change, that means concentration. Okay. So let's look at how the catalyst works.
Reaction to occur, even one that releases energy, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, EA. This required energy input represents a barrier that prevents even energy-releasing reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme's substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction. To the specific fit between enzyme and substrate, each enzyme can catalyze only one kind of reaction involving specific substrates. Thousands of different enzymes may be required to carry out all of a cell's metabolic processes.
So that's the idea with a catalyst, right? A catalyst speeds a reaction up by lowering the activation energy, but doesn't have to rely on an increase in temperature or concentration to get it done. So in a living system, thousands of enzymes catalyze millions of reactions every day.
Even one that releases energy.
Hold on. Okay. Thousands of reactions occur every day in order to give the cell what it needs quickly enough for the cell to function. Okay. So catalyst speeds up a reaction by lowering activation energy. Okay. Makes it easier for the reaction to happen. Now, catalysts are specific and they can be regulated. And here we're talking about biological catalysts, most of which are protein. Now, are there other kinds of catalysts? Yeah, there are catalysts that aren't proteins. There are catalysts that are things like metals that are used in chemical experiments in laboratories, right? The catalytic converter in your car is platinum, and it's designed to completely combust the gasoline that comes out of your tailpipe So the only thing that comes out the back is CO2 and water, as opposed to partially unburnt hydrocarbons, right? That's one of the reasons why you don't leave your car parked in a neighborhood of high crime for a long time, because they'll steal your catalytic converter, let alone the other stuff in your car, because platinum is very expensive. But in living systems, it's proteins called enzymes that do this. Okay? Enzymes. Okay. What the enzyme does is it comes in, it grabs the reactants, forces them to react in a way that it wants them to, and then lets go of the products, and then moves on to the next set of reactants. So it does it very quickly. And enzymes can be inhibited or activated, which means it can be controlled, and that helps the cell make what it needs in the right amount at the right time, right? Some enzymes in living systems are RNAs that can catalyze their own breakdown, okay? But most are proteins. Most catalysts. Living catalysts. Okay. The substrate is just the reactant that specifically is acted on by the enzyme. An enzyme's particular substrates are going to bind the active site, right? Let's take a look at how enzymes work.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
So there it is, right? An enzyme can act only on molecules that fit its active site, just like a key only opens one lock, okay? An active site squeezes the substrates together and stretches bonds so that the bonds are easier to break, or in some cases to make. That lowers the activation energy, and as a result, the reaction happens more rapidly, right? Product leaves the active site after it's finished, and the enzyme can turn around and do the same process again, right? So we can zip up here, whoops, back up. We can zip up here and start all over, okay? So once we get to here, we can zip right back up here, do the same thing again. Until what? Until we have enough of the product that we don't have to keep whipping it out, in which case, generally, that product will attach to another region on the enzyme and reduce its activity until that concentration of that product falls again. And that's called negative feedback, okay? Each enzyme has a set of conditions under which it works best. A proper pH, a proper temperature, a proper salt concentration are critical for the enzyme to maintain its shape in order for it to properly function. And when those conditions change, the enzyme can denature, okay? And if the enzyme denatures, then it won't work anymore. And that's one of the reasons that organisms try to maintain these values whenever possible in a process called homeostasis, in which it uses a series of body systems to try and keep those parameters within a window that's compatible with life. Okay, now in the human body, The communication networks for homeostasis are the endocrine system and the nervous system, okay? And other organisms, there are other body systems that help it to maintain those conditions, okay? Regulatory molecules can speed up or slow down an enzyme by binding to either the active site or to sites other than the active site, okay? So this is an example of something called inhibition. And sometimes activation. Okay. Inhibition can work at the active site or at a different area of the enzyme. The regulatory site, for instance, okay? Activation usually happens at a different region of the enzyme. Okay, all of these involve shape changes. Okay, so an example here, right? You got a regulatory molecule, it can bind to an enzyme, and that can cause it to click on and bind its substrate. So in this case, we're looking at here is activation. Okay, we could have something similar in which you've got an enzyme, okay, a little enzyme, and it's supposed to bind a substrate. Substrate, a different color. Okay. Here's the substrate coming in. Okay. And then it releases. Let's say that it splits the triangle in two. It releases the product. Okay. And then it could go back and start all over again, right? Now, what happens when this concentration gets really high is that this might feed back and inhibit that enzyme so that it slows down or stops, so that we only make what we need when we need to make it, okay? And that's inhibition. In competitive inhibition, you bind the active site and don't let the enzyme proceed. And in non-competitive inhibition, you bind someplace other than the active site and keep the reaction from proceeding, okay? Sometimes enzymes need tools in order to do their work, right? Metal ions or coenzymes associated with an enzyme and are necessary for it to work. Examples include dietary vitamins and minerals. A coenzyme comes from usually from a vitamin, okay? It can be modified by a reaction. And these are things like the B complex vitamins, for instance, or vitamin K, okay? Vitamin E, okay? And what happens is that they participate in the reaction sometimes by moving chemical groups from one molecule to another, okay? Think of them as tools that the enzyme uses to do its work, right? So you get these where? You get these from your diet, okay? All right, we talked about energy, right? And when we're talking about energy, we're talking about the need for something that can be split to release energy immediately, that can be trapped to do something that normally wouldn't happen, like build something or move, okay? And in living systems, that comes from something called ATP. Okay, so let's learn a little bit about ATP here.
Everybody, this is Rob Slotsky from New York campus of PACT, and welcome back to my Bio 121 Anatomy and Physiology 1 course. This is podcast 2.5H, adenosine triphosphate, ATP. The last of the organic molecules that we will discuss in this chapter is adenosine triphosphate, more commonly known as ATP. ATP is the universal form of chemical energy used by cells to carry out energy-demanding cellular activities, such as macromolecule synthesis, active transport of chemicals across cell membranes, movement of cells and cellular structures, and muscle contraction. One way to understand the function of ATP inside a cell is to think of it as a type of energy currency. When a cell needs work to be completed, it must spend ATP, just like we need to pay others money for services performed. ATP is technically classified as a nucleotide having a three-part structure. It consists of a molecule of the nitrogenous base adenine bound to the sugar ribose, which together are called adenosine, attached to three phosphate groups. The term triphosphate comes from the presence of these three phosphate groups. The negatively charged phosphate groups are attached to each other by two phosphate bonds, a type of covalent bond often represented by squiggles in ATP's molecular structure. These phosphate bonds are used to transfer energy that powers endergonic reactions. Remember that endergonic reactions require an overall input of chemical energy. In order to transfer energy from ATP, the third terminal or last phosphate group is removed through hydrolysis, the addition of a water molecule, and there is a net release of free energy that can be used by the cell. The specific enzyme that catalyzes this catabolic reaction is called ATPase. Once the hydrolysis of ATP is complete, the resulting product is called adenosine diphosphate, or ADP, because it contains two phosphate groups. The free phosphate group released from this reaction can now be attached to another reactant or enzyme, or be used to synthesize new ATP molecules. ATP can also be regenerated through a dehydration synthesis reaction using the enzyme ATP synthase and a net input of energy, coming mostly from the breakdown of glucose via cellular respiration. In this reaction, a free phosphate group is added to ADP, which then yields one molecule of ATP and one water molecule as products. Think of the overall hydrolysis and synthesis of ATP as a continuous cycle, where usable energy is constantly consumed and produced inside a living cell. Thanks for watching. Please contact me by email at rjswatsk@hack.edu if you have any questions or comments.
So ATP is a form of energy that's immediately available to do work in living systems. You think of it as biological gas in the tank, okay? When a phosphate group is split off of ATP, the energy released from that chemical bond can be captured and used to do other work that normally wouldn't take place, okay? Just like, you know, if a construction crew is going to build a building, they better eat some food so that they have a source of energy to do the work of putting the building together, all right? Think of it like that, okay? All right, let's pause for a moment and then we'll resume.
Recording in progress.
Okay. All right, so we look at ATP as an energy carrier, and what we see is that these high energy bonds are where the action is. All right, this is essentially where we've put the bulk of the chemical potential energy. Okay. Okay. So when we crack these bonds, the energy comes out and we use it to do work. Now, why are these high energy bonds? Because there's negative charges on each of these phosphate groups, and to hold negative charges together takes a lot of power, which means there's a lot of juice in those bonds, right? So what happens when we hydrolyze ATP is that energy comes out, and then we use that energy to do other work. All right. So that goes off to do other work, right? And then fuel in the form of food, okay, is used to reassemble ATP, right? So energy from food is used to put ATP back together, right? In a process um, that involves either stealing the energy directly from the chemical bonds in the food or using the energy in the food to create what we'll find out in mitochondria is a proton gradient to drive ATP production. And we'll get into those details a little bit later. Okay. Cells concentrate, convert, and dispose of most substances in enzyme-mediated reaction sequences. And the metabolic pathway is simply a series of chemical reactions that allows that to take place, right? So you've got an initial reactant. There's an enzyme that converts that into an intermediate, another enzyme that converts that into another intermediate, and so on. And then eventually you get to the product. And then usually when the product builds up to a certain point, it will feed back to the first enzyme in the pathway. Actually, let's put this up here. And that will slow the reaction down. Okay. So, like that. Okay. And that's so that we have control. Very important. Okay. A linear pathway goes straight from a reactant to a product, while a cyclic pathway um, ends up with the last product being the reactant of the first reaction. Okay. So around and around and around we go. And then the last reactant is the substrate for the first enzyme. Okay. So it's the reactant for enzyme 1. Right. But also the product of enzyme 3. That's why it's a cycle. Cyclic, that's linear. Okay. We can control metabolism by slowing down reactions that have already filled the bill for what we need. Okay. We can either do competitive or non-competitive inhibition. Generally, the concentration of the final product in a chemical reaction series, a metabolic pathway, feeds back to some of the earlier enzymes in the pathway and slows down the activity, just like shown here. Okay. So there we are, right? And that way we don't waste our time and our energy. Now, one of the ways that energy is transferred is by moving electrons from one location to another. Okay. When electrons change their location, they change their potential energy, right? Just like if I took the elevator from the 5th floor to the 4th floor, my potential energy would be reduced because I'm not as high above the ground anymore. Okay. So in an electron transport chain, what happens is that we change the position of electrons. Basically, what we do is we produce a current in which electrons go from high to low energy. And that current drives work used to do other things. Okay. We're going to find out that in living systems, okay, the part of the metabolic pathway that makes the most ATP, which is called the electron transport chain, okay, relies on two currents, right? One runs from NADH and FADH2 all the way to oxygen, with carriers helping it along the way, right? And so this is a current of electrons, okay? Remember that a current is just a moving charge, right? And that energy is used to make another current, which is a proton current, which moves from the matrix or from the intermembrane space to the matrix of the mitochondria across the membrane, okay? This would be the inner mitochondrial membrane, right? And that's used to produce the bulk of the ATP from the ADP and phosphate. Okay, so that also is a current, okay, moving charge. So two currents that drive that production. We want to do that in a stepwise fashion in living systems, right? So that we don't generate so much heat that we burn up the organism, right? You see an example here, right? This is a one-step oxidation of glucose. You see the fire, right? That's not going to work with life, right? Whereas a multi-step oxidation of glucose releases heat in a stepwise fashion so that we don't cook the organism, right? And when we're talking about oxidation, we're talking about stripping the energy from glucose to make ATP, okay? So I'm just setting you up for the next chapter. So one of the things we have to appreciate about living systems is not only do they have to take energy from fuel and use that to do work, but they also have to determine what they want to let in and let out of the cell in order for it to function. Okay, so there's two types of cell transport that sustain the cell, and those are passive transport and active transport. Okay, active transport requires an energy input, and passive transport doesn't require an energy input. Okay, so two very different setups. Right? So under passive transport, let's look first at diffusion.
Molecules dissolved in a solution are.
Hold on. Let's do this.
Molecules to molecules to
Why is it like that? There we go. again.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. Lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. Type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Okay, so with diffusion rate, remember this is a form of passive transport. This is going to depend on several factors that are going to dictate the speed with which this takes place. Okay, and those factors are concentration, temperature, size, charge, and pressure. Now, how is it that those are affected? Well, the answer is fairly straightforward. Okay, if we look at, and let's engage the pen here. Come on, let's go here. Come on. There we are. Okay. So, the way this is going to work, right, is that the higher the concentration, the greater the speed. The greater the temperature, the greater the speed. The greater the size, the slower the speed. Okay. The greater the pressure, the greater the speed. Okay. And the charge depends on whether the charge is positive or negative with respect to the available area. Okay, but if you've got similar charge, the more charge you have, the more rapidly it's going to spread out. Okay. All right. So when fluids on either side of a selectively permeable membrane differ in solute concentration, water diffuses across the membrane in a direction that depends on its tonicity, right? Hypotonic solutions have a low solute concentration relative to another fluid. Hypertonic, high solute concentration relative to another fluid, and isotonic have the same solute concentration. Okay, so what we're talking about here is in relationship to a point of reference, right? If we have, let's go back for a second, okay? Let's say that we have a cell, okay? So we'll draw a cell in three separate situations, okay? A cell here, here, and here, okay? We'll put a nucleus in there, okay? All right. Now, we'll call this one isotonic, this one hypertonic, and this one hypotonic, okay? And then we'll use different color for the water, so let's do that. Water is blue in this illustration. Okay, so we've got a cell in a hypotonic solution here in a container, right? And an isotonic solution here in a container, and in a hypertonic solution here in a container. Okay, and then let's indicate the solutes in black. Okay, solutes are just materials that are dissolved in the solution. Okay, so in a hypertonic solution, there's lots of solute outside the cell. So there's more water per unit area in the cell than outside the cell. Okay, in an isotonic solution, so there's maybe a few solutes here. In an isotonic solution, the amount of solutes is approximately the same inside the cell and outside the cell. Okay, in a hypotonic solution, there's more solute inside the cell than outside the cell. So the question we want to ask is, what is the direction in which water is going to move? Okay, so to see that, let's use blue arrows for water. Okay, all right. In a hypotonic solution, water is going to enter the cell more rapidly than it will leave. Okay, so water's still going to flow in both directions, but more water is going to flow in than flow out. Isotonic solution, you're going to have equal movement in either direction. And in a hypertonic solution, more water is going to flow out than is going to flow in. Okay? And by water, we mean free water, water unoccupied with any other activity. Okay? So, what's the result going to be? Well, result, we'll put that in green. Okay? In an isotonic solution, no change. In a hypertonic solution, shrivel up, or we could say dehydrate, or crenate if you like. Okay? And in a hypotonic solution, it's going to lyse or burst. Okay? So this is why an organism is going to try very hard to maintain an isotonic condition inside and outside the cell. Now the lipid bilayer is selectively permeable, okay, meaning what? Only certain things can go through freely, okay, and those are any hydrophobic molecule and small polar molecules, okay. Water would be one of those unusual exceptions, although a lot of water goes through special water pores called aquaporins. And those are actually protein tunnels, okay. Anything charged or polar isn't going to go through, all right. What this does is allows the cell to dictate what can move through and what can't. That's very important because that allows the cell to dictate its internal conditions, okay. Not just anything can go through the cell. Why can't charged and polar things go through the phospholipid bilayer? Because the hydrophobic core of the bilayer makes too big of an energy barrier for that to happen. So the only way that ions are going to go through is with the aid of a gate or a tunnel or a pump. Okay? And those are made out of protein. Okay? All right. Next topic here is osmosis, which is simply the diffusion of water. right? So let's take a look at how osmosis affects a cell, and it's going to affect it the way that we indicated further, okay? Hypertonic and hypotonic solutions. Hypertonic causes the cell or the system to shrink. Hypotonic fluid will cause it to swell. Isotonic, no change, right? So let's take a look at osmosis.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Let's look how this works in red blood cells.
The image on the left is human erythrocytes, or red blood cells. The image on the right is enhanced with filters. These living cells are in isotonic solution. Note the lozenge shape. Now the solution has been changed to have a high salt concentration. It is hypertonic. The red blood cells shrivel and become crenated. Finally, the solution has been changed to plain water. The cells swell and burst, leaving empty membranes behind. This phenomenon is called hemolysis.
So this is one of the reasons why, if you are NPO, okay, NPO, let's define that. And there we go. NPO means equals nothing by mouth. And you are dehydrated. Okay. We don't give you an IV of distilled water. Instead, we give you IV of isotonic saline. Okay. If it's isotonic, the red blood cells aren't going to rupture because the rate at which water moves in will be equal to the rate at which water moves out, and the cells will maintain their normal shape and function. If we were to give you pure water, we would rupture the cells, right? They would release their hemoglobin. The hemoglobin would block capillary beds and cut off blood to downstream tissues, and those tissues would die, okay? So now you have a practical application for what you've learned here with membrane transport, right? In plant cells, turgor counters osmosis. Turgor is the pressure that a fluid exerts against a wall, a membrane, or another structure that contains it. Osmosis continues until two fluids are isotonic or until pressure against the hypertonic fluid counters that movement, okay? So you can see here the effects of tonicity in plant cells, okay? On the left, turgor pressure is high in these cells of an iris petal. They are plump in the cytoplasm, while turgor pressure on the right is low in those cells of a wilted iris petal. The cytoplasm has shrunk and the plasma membrane is pulled away from the cell wall. So you see here the purpose for the cell wall, right? The cell wall protects against lysis. All right. And that is in any organism that has cell walls. So those would include fungi, plants, many bacteria, okay, and some protists. And some archaea. Okay. Gases, water, and small nonpolar molecules can diffuse across a lipid bilayer without any help. They just move down their concentration gradient from high to low. Most other molecules and ions cross only with the help of some sort of a protein in the membrane. Okay. So in facilitated diffusion, for instance, we're going to find out that you need a helper protein in the membrane that ferries you across if you're charged or polar, right? Each type of transport protein moves a specific ion or molecule across the membrane. If it's passive transport, it moves down the concentration gradient. If it's active transport, it moves against the concentration gradient. Okay? The concentration gradient in passive transport always determines the direction of movement from high to low. So it requires no energy input. Example would be a glucose transporter, all right, where glucose would move into the cell from high to low, okay. In active transport, we use a source of power in order to move material against its concentration gradient, such as ATP in primary active transport or in vesicle transport, or we can use a gradient set up by primary active transport to drive something called secondary active transport. So we can see some examples here, right? With facilitated diffusion, right, a glucose transporter can bind high concentrations of glucose on one side of the membrane, okay, and then pass it into the cell, all right, via facilitated diffusion, okay, from high to low, all right. Calcium pump, right, is going to move material against its concentration gradient using ATP to drive it. Okay, so this is called primary active transport. Okay, right, uses ATP. Okay, and then in secondary active transport, you use a gradient set up by primary active transport to do the work. Now, vesicle transport is a type of active transport that requires ATP to move membrane-enclosed vesicles from one location to another. In endocytosis, the cell membrane pulls something in from the outside. Could be a solid, could be a liquid. If it's a solid, we call it phagocytosis, okay? And if it is a liquid, we call it penocytosis. Okay? Penocytosis. That's an I there. Liquids. Okay. And in membrane bound endocytosis, you can also have receptors that bind particular molecules and pull them in, and that's called receptor-mediated endocytosis. Okay, so you've got variations on a theme here. But they're all active transport because they all require ATP to move these vesicles along microtubules that are inside the cell. So the little vesicles literally walk along the microtubules like a train moving down train tracks. In exocytosis, the cell manufactures something and then kicks it out of the cell, okay? But it's still a form of active transport. So let's take a look at this more closely.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use three basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
So you can see some more examples here. Here you're looking at some more active transport mechanisms. The sodium-potassium pump is an example of an antiporter. Okay, antiporter. Means moves two materials in opposite directions. Against their concentration gradients. The square brackets mean concentration. And the way that this works is that a single ATP is used to drive the movement of 3 sodium ions out of the cell for every 2 potassiums it drives into the cell. And this is an example of one of the important principles in the cell, which is called membrane potential. Okay, this is a separation of charge across the membrane that serves as an energy source. Okay, so the cell uses this to pull materials that it needs in and kick material that it doesn't want out so that it doesn't die. Okay, an example of a material that the cell needs to pull in, often against its concentration gradient, is glucose, right? And the sodium glucose symporter relies on the sodium gradient set up by the sodium potassium pump in order to pull the glucose into the cell. Okay, so without the sodium potassium pump using ATP, there would be no way for it to, the cell, to grab glucose from outside and pull it in against its concentration gradient. Okay. So let's look at all of these membrane transport mechanisms together.
In order for us to use the molecules and energy we get from food, we must transport them across cell membranes. Transport of nutrients is essential for all cells, but we'll use cells in the small intestine to teach the principles of membrane transport. Let's zoom into the surface of an intestinal cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can move freely across the lipid bilayer. Notice that the lipid molecules move down their concentration gradient from where they are more concentrated to where they are less concentrated. This is an example of simple diffusion. Diffusion is a form of passive transport. It does not require energy from the cell, but it does depend on the concentration gradient, size and charge of the molecule or ion. However, most molecules cannot cross the lipid bilayer. Instead, molecules like the sugar fructose move into intestinal cells by facilitated diffusion, moving down their concentration gradient and through transport proteins. It also differs because it acts only on specific molecules, and the transport proteins can become saturated, reducing the amount of diffusion. However, facilitated diffusion doesn't require energy from the cell, so, like simple diffusion, it's a form of passive transport. The majority of water molecules cross the plasma membrane by facilitated diffusion, also called osmosis, through specific protein channels called aquaporins. Some ions, such as sodium and potassium, require transport against their concentration gradients. The sodium-potassium pump actively transports these ions from where they are less concentrated to where they are more concentrated. This process requires energy from the cell and is known as active transport. Energy from ATP is used to move 3 sodium ions, shown in red, out of the cell, and 2 potassium ions, shown in blue, into the cell. Because the pump uses ATP directly, this type of transport is called primary active transport. Another type of active transport, secondary active transport, also moves chemicals against gradients. It doesn't use ATP as a direct source of energy. Instead, it uses the downhill gradient from one ion or molecule to drive the uphill movement of another ion or molecule. For example, sodium and glucose move into the cell by secondary active transport in a process called cotransport or symport, because both substances are moving in the same direction. Sodium ions move down their concentration gradient created by the sodium-potassium pump, but because glucose concentrations inside the cell can quickly exceed those outside the cell, glucose moves against its concentration gradient. The cell moves larger materials, such as proteins, from the intracellular fluid to the extracellular fluid, using vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. Cells use all these processes, passive and active transport, exocytosis and endocytosis, to move materials across their membranes.
So you can see, again, that this is a combination of mechanisms, because sometimes the cell has to pull stuff in against its gradient or push stuff out against its gradient, or it has to work with the gradient in order to move stuff in and out, okay? So in simple diffusion, we're talking about the movement of nonpolar molecules from high to low concentration, either into or out of the cell. It doesn't require a membrane protein to happen. It happens on its own. With osmosis, we're talking about the diffusion of water across a semi-permeable barrier from high to low free water concentration, right? So the way to think of osmosis, the easiest way that I remember to think about it, is water chases solutes, okay? So let's write that down real quick, okay? Water chases solutes. Okay. Facilitated diffusion is movement through protein channels in the cell membrane. Active transport, movement across membrane against concentration gradient requiring ATP, okay? And then you've got, with active transport, vesicle transport, and secondary active transport. Active transport. Okay. In secondary active transport, a primary active transport gradient is used to drive movement of another solute against its gradient. Okay, and with vesicle transport, ATP is used to move material into or out of the cell and it's active transport because ATP is required, okay? And this way that we move it is in a vesicle, okay? All right, so some points to ponder, right? Testosterone supplements have been used by athletes for some time. Why do the athletes use them? Why use testosterone instead of estrogen? Should the cause of liver failure be used as a factor to prioritize liver transplants? What is the alcohol flushing reaction? Why does it occur when drinking alcoholic beverages? Okay. So, you know, let's give you some clues here. Okay. Anabolic steroids. Okay. I want you to look that term up and understand what it means. Okay. Testosterone is an anabolic steroid. All right. All right. The cause of liver failure. Right. Why does liver fail? Okay. Well, blanket term is hepatitis. All right. But that's a blanket term meaning inflammation of the liver. The liver, right? And there's a variety of reasons the liver gets inflamed. Sometimes there are viruses that do it. Sometimes there are chemicals that do it. Okay. Sometimes it's related to exposure to certain toxins. Okay. So you want to think about the cause of the hepatitis, right? What is the alcohol flushing reaction? Okay. So I want you to look up what is ADH. What is ADH? Okay. And how does it control water balance? Okay. All right. That brings this particular chapter to a close. We're going to talk about this lecture in class. We'll do Q&A over it, right? But what I want you to do is to take notes as if you were at this lecture live, and then when we get into class, we do Q&A over what you've learned, and then we try to probe deeper understanding, all right? I appreciate y'all for listening.